I'm Katrina Lythgoe and I'm an evolutionary epidemiologist here at the Big Data Institute in Oxford. During the pandemic, working from home has actually really had its challenges because a lot of science comes from those spontaneous conversations that you have in the coffee room, over lunch, in face-to-face meetings. It's a great feeling to be doing work that you know could directly save a lot of lives, but at the same time there's no getting away from it. When your work is coronavirus and your friends are all asking you about coronavirus, it can all get quite overwhelming quite quickly. Coronaviruses are RNA viruses, and they're essentially like packets of information encoded in the RNA, which is then enclosed in a particle. Those particles need to infect host cells in order to replicate, leave the host cell, and then go on to infect more cells within the body. The coronavirus RNA, we can imagine it as a string of 30,000 letters, and through genetic sequencing, we can read those letters across the genome. What we find is as the virus spreads from person to person, we can start to see mutations emerging in some individuals and then spreading into other individuals, which then go on to create this kind of evolutionary change. By looking at those changes, we can map how the virus is spreading within countries and also globally.
In the early stages of the pandemic, a mutation in the genetic code of the coronavirus occurred inside the body of a single infected individual. This created a new variant called D614G. This variant established dominance as the most common form worldwide and was responsible for over 85% of new COVID-19 cases by summer 2020. Because viral evolution is an ongoing process, more variants will likely emerge that improve the virus's ability to transmit and spread. In late 2020, another new variant was detected in the UK, with mutations that increased transmission of the virus.
Looking to the future, we'll be on the lookout for mutations that, for example, could render vaccines less effective. One thing that is on our side with coronavirus is that it evolves extremely slowly, and therefore it will make it harder for the virus to escape immune responses. So we're hopeful that vaccines will be effective in the long term.
Katrina and her team are also exploring how the diversity of the virus within an individual can give us insight into detailed patterns of spreading.
Because of this replication of the viruses and its spread within the body, you can have millions and millions of viral particles within a body. And when we sequence the virus, we're trying to sequence as many of those virus particles as we can. Generally, what people do with that information is they'll take the average genetic sequence to create what we call the consensus sequence. We can think of a metaphor, for example, of people in a city. So let's imagine everybody wears a white t-shirt and black jeans. The consensus in that city will be a white t-shirt and black jeans. But then you may have somebody who comes along and decides to wear a pink t-shirt, and their group of friends also wear pink t-shirts. So the consensus fashion, if you like, will still be white t-shirt and black jeans, but there will also be a proportion of individuals that are wearing a pink t-shirt. And we're interested in understanding that diversity, so not just the consensus, but also those individuals with the pink t-shirts, if you like. Of course, that's a very simplified example because the coronavirus genome has about 30,000 base pairs, so there's 30,000 places where you could see a change in the virus. So that's a different t-shirt, that's different jeans, that's different shoes, that's different socks, and so on and so on. It becomes extremely complex very quickly. But with modern sequencing technology, we really can be very confident that we can follow those individual changes.
This sort of high-resolution genetic detail offers a vital understanding of viral transmission within small clusters of people.
We can think of households, and we may want to understand who was the first person to bring the virus into households. Does that tend to be school-age children, for example, who are then infecting their parents? Or is that parents who are coming back into the household and then infecting their children? And then this can have quite important implications for intervention strategies. There's been a big shift in our priorities during the pandemic, because before the pandemic we may have been trying to understand long-standing problems, whereas now the priority is about public health benefit as fast as possible, whether that will lead to a publication or not. There really has been this feeling of mobilisation, so I'm now collaborating with people that I never would have collaborated before to bring our kind of shared expertise together to really try and understand the virus and do everything we can to control its spread.